Scientists have found that non-coding junk DNA, far from being harmless and inert, could potentially contribute to the development of cancer. Their study has shown how non-coding DNA can get in the way of the replication and repair of our genome, potentially allowing mutations to accumulate. It has been previously found that non-coding or repetitive patterns of DNA, which make up around half of our genome, could disrupt the replication of the genome. But until now scientists have not understood the underlying mechanism, or how it could contribute to cancer's development. In the new study, scientists at the Institute of Cancer Research, London, reconstituted the entire process of DNA replication in a test tube in order to understand it more completely. The researchers were able to describe how repetitive patterns of DNA are copied during replication and how they are able to stall replication entirely, increasing the risk of errors that can be an early driver of cancer. The researchers believe the work could also help to improve the diagnosis and monitoring of some cancers, such as bowel cancer, where common errors in copying the repetitive sequences of DNA indicate whether cancer is progressing. The study, published in Nature Communications, was funded by Wellcome and the Royal Society, with additional support from the Institute of Cancer Research, ICR, itself. Scientists at the ICR, a charity and research institute, found that when the DNA replication machinery encountered repetitive DNA, it was able to unwind the DNA strands, but it sometimes failed to copy the opposite DNA strand. This error could cause replication to stall, resulting in collapse of the replication machinery in a manner similar to that induced by DNA damage. DNA damage and ensuing genome instability are known to promote cancer formation and progression, so the research strengthens the link between junk DNA and cancer. It was scientists at the ICR who, in the 1960s, provided the first conclusive evidence that DNA damage is the fundamental cause of cancer. In the early 2000s, ICR researchers then showed that drugs called PARP inhibitors could be genetically targeted against cancers with DNA repair mutations. Our researchers now hope that improved understanding of DNA replication, and how it can go wrong, might lead to new ways of treating the disease. Our study suggests that so-called junk DNA is actually playing an important and potentially damaging role in cells, by blocking DNA replication and potentially opening the door to cancerous mutations. Our study therefore fundamentally advances our understanding of cancer, and I'm hopeful it will help us come up with new treatments in the future. Professor Christian Halen, Chief Executive of the Institute of Cancer Research, London, said, "This study helps to unravel the puzzle of junk DNA, show. It's possible that this mechanism could play a role in the development of cancer as a cause of genetic instability, especially as cancer cells start dividing more quickly and so place the process of DNA replication under more stress." Understanding the mechanisms underlying genetic mutation and instability is critical if we are to find innovative new ways to treat cancer that exploit fundamental weaknesses in cancer cells. Corella S. Casas de Luque, Manuel Daza Martin, Sophie L. Williams, Gideon Koster. The mechanism of replication stalling and recovery within repetitive DNA. Nature Communications, 2022. 13, 1, doi, 10. 1038 per second 41467, 02231657 X Institute of Cancer Research. Junk DNA could lead to cancer by stopping copying of DNA. Science Daily, the 19th of July 2022. ift.tt/s0cgkdx. Institute of Cancer Research. 2022, July 19. Junk DNA could lead to cancer by stopping copying of DNA. Science Daily. Retrieved July 19, 2022 from ift.tt/s0cgkdx Institute of Cancer Research. Junk DNA could lead to cancer by stopping copying of DNA. Science Daily. ift.tt/s0cgkdx. Accessed July 19, 2022. Thanks for watching. Please subscribe my channel for more videos.